Hello, girls. So, today we're going to talk about Chapter 23, the chapter on the evolution of populations, or what we might call microevolution. And so with microevolution, we're focusing on the population level, the evolution of groups of individuals of the same species. And it's a change in that population, that species, from one generation to the next. As I've said, that population are individuals of the same species that are in a given area, and they have a particular gene pool or combination of genes in that population. And you can have different populations of the same species, obviously, with some overlap, maybe not overlapping. Hardy-Weinberg is a mathematical description of populations and their evolution through time. We're going to work on it in that lab. What it says is that given certain conditions, the allele and genotype frequencies in populations do not change from one generation to the next, and we'll see what those conditions are in just a minute. All right. So again, this one describes a population and its changes through time. It requires, as we'll see, random mating. And again, under those certain conditions, it describes a population that's not changing. Here's the Hardy-Weinberg equation. And these represent alleles. P is typically represents, say, a dominant allele, and Q a recessive allele. P squared and Q squared represent the homozygous genotypes, whereas PQ represents the heterozygous genotype. And you can see the frequency of all these genotypes and their combinations within the population is going to be equal to a frequency of 1. All right. And so you notice in the equation it's 2PQ representing the heterozygous genotype, and the reason you put a 2 there is because there's two different ways, two different combinations under which you can get that heterozygous condition, depending on what you get from mom and dad. There's only one way you can be homozygous dominant or homozygous recessive, and that's if you get the homozygous dominant allele from mom and dad, and the same with the homozygous recessive. But there can be situations where you get the recessive allele from one parent or the recessive allele from the other parent, and vice versa. So again, this describes, it's a mathematical description of a population, a hypothetical description. And it says that allele and genotype frequencies do not change over time, but obviously those allele and genotype frequencies do sometimes change. Now these are the conditions under which allele and genotype frequencies will not change. When you have large populations with little or no gene flow, that is, genes moving in or out of the population, no mutations, no new alleles showing up, random mating within that population, and basically no selection. What this says is this applies to a population that is not evolving. All right, now in terms of the gene pool, what can change the gene pool? Mutation obviously can contribute new alleles, and recombination can put alleles and genes in new combinations. And as we learned in the genetics section, mutations come in all kinds. You can have very simple point mutations where you get single nucleotides changing, which can create, again, new alleles that generates variation like in these horses. Sometimes, as it says here, these are harmless, those silent mutations, but other times they can have an effect on the phenotype. And we saw you can get larger mutations at the chromosome level. These often can be, as it says here, quite harmful when you either have extra pieces or you're missing pieces of your chromosomes, for example. Mutation rates, I don't know, relatively low, but of course, with a large enough population and enough time, mutations can appear and accumulate in a population. And sexual reproduction and the recombination that occurs there through meiosis and fertilization puts those alleles and those genes in unique combinations each generation, creating variation. Okay, so what can cause microevolution to happen? What can cause allele frequencies to change through time? Well, these are the three things. Selection can clearly do it when over time particular genetic combinations, particular alleles are being favored. That will cause those to increase in frequency and others to decrease in frequency. So from one generation to the next, you can get, again, a change in allele frequencies and genotypes. Genetic drift, this is a phenomenon that occurs generally in small populations, populations that become small, when you can get sort of random, unpredictable changes in that population. When populations decrease in size, it tends to reduce, as is probably self-evident, genetic variation. And so this is a cartoonish example of this, where we've got one population, but again, it's relatively small, and just due to the randomness of mating, you can over time get a significant change in allele frequencies. This genetic drift is sometimes referred to as resulting from what's called the bottleneck effect. And that's when you have a relatively large population that over a short period of time decreases significantly in size, as is shown here with this cartoonish image above, where we have this large population with several different genotypes and different particular frequencies. Well, after the population goes through a bottleneck and decreases significantly in size, that subsequent population, the surviving population, as shown here, may not be representative of that previously large population. You've lost certain alleles and genotypes. And we have the picture of the elephant seals here, is because they went through a bottleneck more than a century ago, in which in the 1800s their populations were in the millions, but then they were hunted nearly to extinction, only a few thousand individuals remaining. And the genotype frequencies, the diversity in these populations now are relatively low, even though they have rebounded some. There's not a whole lot of diversity there. Also, the founder effect, when you take individuals from a large population and they migrate to an area, they will not necessarily be representative of that whole population. Gene flow can modify allele frequencies and genotypes. When you have genes moving between populations, immigrants can bring in new alleles, new genotypes. Individuals leaving a population can cause the loss of alleles and genotypes. With enough gene flow, as you can imagine, populations tend to become less differentiated over time because their genotype and allele frequencies tend to become very similar with, again, enough migration. All right, natural selection operates on genetic variation. You have to have some variation really for evolution to occur. Ignore that. Let's see. So there can be different types of variation, discrete and quantitative. We've talked about this a little bit already. First semester, again, discrete, single gene traits, quantitative characteristics, multi-gene traits. Again, these all can vary within a population, leading to polymorphism, and that's just where you have different forms, different morphs within a population. How do we measure genetic variation? Well, you look at genotype frequencies basically, and often you talk about levels of heterozygosity. How heterozygous is this population versus being homozygous? And okay, again, with between populations, you often have variation, and again, depending on the level of gene flow between them, they can be not that different or really quite different. If these mice are separated by a mountain range or something and there's very little gene flow between them, they'll be quite different. Variation can differ along what's called a cline or across a transect through space, depending on the habitats. You can see plants that are in more lower elevations in coastal regions might have particular genetic combinations that are different from those found at higher elevations, because the genes that help them survive at higher elevation will be somewhat different than the ones that help them survive at lower elevation. They're the same species. Again, you just get genetic variation based on habitats that they're found in. Okay. All right. So when we speak of survival of the fittest, these kinds of terms, by fitness, what we mean is what's sometimes referred to as Darwinian fitness, and that is how successful are you at reproducing, at passing on your genes to the next generation. And so the most fit individuals, in a Darwinian sense, pass on the most copies of their genes, and they have high genetic or high Darwinian fitness. Okay. So if we talk about the relative fitness, what we mean there is the fitness of particular genotypes. Are particular genotypes more successful at passing on their genes to the next generation than other genotypes? Selection. So we're doing our experiment on artificial selection, and we're doing what we would call directional selection. We're taking a particular population and driving it in a particular direction. And natural selection can operate that same way. But you can also have disruptive selection, where a population tends to split into two different types, or stabilizing selection, where the average type is favored. And so here's the image again. Directional selection. Over time, the phenotype is shifting towards one end of the phenotypic spectrum. Disruptive selection, where the population again is sort of being split into two different forms, two different morphs. They may occupy slightly different habitats where either a darker or a lighter form is favored. And so therefore you have, again, disruptive selection. Stabilizing selection is when an average type is favored. In people, we can think of over evolutionary time, and in our lineage there's been directional selection for having bigger brains, for example. With stabilizing selection in birth weight of babies, there's been stabilizing selection towards about 7 pounds or so, because before modern medicine, if babies were too small or too large, they tend to not survive birth. Okay, preservation of genetic variation. What can keep genetic variation within a population? The diploid condition. When species have 2 copies of each chromosome, they will have 2 copies of each gene, 2 alleles, and that can help maintain genetic variation. Particularly think of recessive alleles. They can just sort of hide out and be masked by dominant alleles and not be selected against. They're just sort of there in that diploid, that heterozygous condition. Balancing selection, there are situations where you can have selection favoring 2 different types, like with disruptive selection, for example. Heterozygote advantage. There are certain situations where heterozygotes have an advantage, again, favoring the maintenance of those 2 different alleles. A good example of that is with sickle cell. Sickle cell is a single gene trait in which you can be homozygous for the sickle cell and you have sickle cell anemia. You can be homozygous not for sickle cell and you don't have it, and you can be heterozygous in which you have a milder form of the disease, and in particular in regions where there are high rates of malaria, those heterozygous individuals are favored because they are somewhat immune to malaria, whereas the homozygous recessives, the ones who do not have the sickle cell allele, will get malaria, and the ones that are homozygous dominant for the sickle cell will have sickle cell anemia. But those heterozygotes have an advantage here because they don't have full-blown sickle cell, and they're also somewhat immune to malaria. Heterozygote advantage. And there you go. Frequency-dependent selection. This can maintain genetic variation because think about it, if a predator is looking for a certain prey, it'll go for the most common form, what is easiest to find, and so therefore the least common genotypes and morphs can be maintained by not being preyed upon so much because they're not as common. So the more frequent you are, there tends to be a little more selection against you under that situation. Okay, I am running out of time, so I'm going to finish this in just a second.